After going over the important aspects of lipid metabolism, let's now discuss amino acid metabolism, which is another high-yield topic to know for test day. Let's now turn to a discussion of amino acid metabolism. Both the protein obtained from the diet or from ubiquitous stores within the body may be utilized as energy sources. Amino acids released from proteins are catabolized primarily in the muscle and in the liver. They usually lose their amino group through transamination or deamination. The carbon skeletons can be converted in the liver to glucose, acetyl-CoA, and ketone bodies. We'll begin by covering the topic of amino acid metabolism with discussion of the removal and excretion of amino groups. Nitrogen in the form of ammonia lacks energy, and the strategy of the body is, if it doesn't need it, it's to eliminate it, and it eliminates excess nitrogen in the urine. The kidneys add small quantities of ammonium ion to the urine, in part to regulate acid-base balance, but nitrogen is also eliminated in the process. Most excess nitrogen is converted to urea in the liver and goes through the blood to the kidneys, where it is eliminated in the urine. Amino groups released by deamination reactions form ammonium, which, if allowed to escape unaltered into the blood in significant quantities, has toxic effects on the brain. Because ammonia would increase the pH of the bloodstream, most tissues add excess nitrogen to the blood by attaching ammonia to the gamma carboxyl groups of glutamate, forming glutamine, which does not change the pH of the bloodstream. Muscle sends nitrogen to the liver as alanine and smaller quantities of other amino acids in addition to glutamine. Most tissues utilize the amino acid glutamine as a major carrier of excess nitrogen. As such, they contain the enzyme glutamine synthetase, which captures excess nitrogen by aminating glutamate in order to form glutamine. Glutamine is released from cells into the bloodstream. It is taken up in the kidneys, which contain the enzyme glutaminase, which deaminates glutamine. This reaction yields an amino group, which is converted to ammonium and eliminated in the urine. The reaction also yields glutamate. Glutaminase in the kidneys is in fact induced by chronic acidosis, whereas the excretion of ammonium becomes a major defense mechanism. The liver has only small quantities of glutaminase, while the levels of this enzyme are high in the intestine, where glutamine is produced from the proteolysis of dietary protein, and the ammonia produced by intestinal bacteria requires deamination. This contributes to the intestinal ammonia entering the portal blood sent to the liver for production of urea. Now let's discuss the role of aminotransferases. Both muscle and the liver have aminotransferases, which, unlike deaminases, do not release the amino groups as free ammonium ions. This class of enzymes instead transfers the amino group from one carbon skeleton of an amino acid to another, usually involving alpha-ketoglutarate, a citric acid cycle intermediate. Aminotransferase requires pyridoxal phosphate, derived from vitamin B6, to mediate this transfer. Aminotransferases are named according to the amino acid donating the amino group to alpha-ketoglutarate. Two important examples are alanine aminotransferase, also known as ALT, and aspartate aminotransferase, also known as AST. Although the aminotransferases are in the liver and muscle, in pathologic conditions these enzymes leak into the bloodstream, where they are useful clinical indicators of damaged liver or muscle. The reactions catalyzed by aminotransferases are reversible and play several roles in metabolism. During protein catabolism in the muscle, they move the amino groups from many different amino acids onto glutamate, thus pooling it for transport. A portion of the glutamate may be aminated by glutamine synthetase, or may have an amino group transferred to pyruvate using ALT, forming alanine. In the liver, ALT and AST can move the amino groups from alanine released into the serum from muscle to aspartate, a direct donor of nitrogen into the urea cycle. The next important enzyme involved in amino acid removal and excretion is glutamate dehydrogenase. It is found in many tissues where it catalyzes the reversible oxidative deamination of glutamate. It produces the citric acid cycle intermediate alpha-ketoglutarate, which serves as an entry point to the cycle for a group of glucogenic amino acids. Its role in urea synthesis and nitrogen removal is still controversial. Let's turn to a discussion of the high-yield urea cycle. The urea cycle, like the citric acid cycle, acts catalytically in that small quantities of the intermediates are sufficient to synthesize large amounts of urea. Urea itself contains two nitrogens, one from aspartate and the other derived from carbamoyl phosphate, both of which are synthesized in the liver. Carbamoyl phosphate is produced from ammonium ions and carbon dioxide by mitochondrial carbamoyl phosphate synthetase. This enzyme is activated by N-acetylglutamate, which is produced only when free amino acids are present and functions as an obligate activator, meaning it is required to turn the enzyme on. The urea cycle occurs partially in the mitochondria and partially in the cytoplasm. Aspartate enters the cycle in the cytoplasm and leaves the cycle as fumarate after donating its amino group. If gluconeogenesis is active, fumarate can be converted to glucose. The product urea is formed in the cytoplasm and enters the blood for delivery to the kidney. It is important to note that urea does not change the pH of the bloodstream and is permeable to membranes, meaning there is no need for transporters. Also, urea is sufficiently polar and thus there is no need for a carrier for it when it is in the bloodstream. Genetic urea cycle disorders are characterized by a combination of hyperammonemia, elevated glutamine, and decreased blood urea nitrogen, or BUN levels. Hyperammonemia can result from genetic urea cycle disorders, which leads to primary hyperammonemia, but it can also arise secondary to other enzymatic disorders which do not directly involve the urea cycle. Hyperammonemia results in excessive amounts of ammonium ion, which depletes alpha-ketoglutarate, inhibiting the TCA cycle. Symptoms of hyperammonemia can include tremor, slurred speech, somnolence, vomiting, cerebral edema, blurred vision, and seizure. The treatment includes IV benzoate or oral phenylbutyrate, which effectively lowers serum ammonia levels by binding amino acids, or lactulose, which binds ammonium ions in the gut so that it is not absorbed by the body and is instead excreted. When ammonia levels are excessively elevated, hemodialysis may be warranted. There are two genetic deficiencies involving the mitochondrial enzymes within the cycle: carbamoyl phosphate synthetase 1 deficiency and ornithine transcarbamylase or OTC deficiency. In both of these conditions, infants typically appear normal for the first 24 hours. During the 24 to 72 hour postnatal period, however, symptoms of lethargy, vomiting, and hyperventilation begin, and if not treated, the neonate progresses to coma, respiratory failure, and death. They can be distinguished by their levels of orotic acid in the blood and urine, which are increased in OTC deficiency, but not in carbamoyl phosphate synthetase 1 deficiency. The reason is that in OTC deficiency, carbamoyl phosphate accumulates in the mitochondrion and spills into the cytoplasm, where it enters the pyrimidine synthesis pathway, resulting in an excess orotic acid and uracil synthesis. The orotic acid is not needed and results in orotic acidemia and aciduria. Both of these conditions are treated with a low-protein diet and the administration of sodium benzoate or phenylpyruvate to provide alternative routes for capturing and excreting excessive nitrogen. Arginase deficiency is also a disorder of the urea cycle, preventing the conversion of arginine to urea. This usually manifests around the age of 3, with progressive spastic paresis of the lower extremities, ataxia, mental retardation, and if severe, signs and symptoms of hyperammonemia. Labs will show elevated arginine with elevated ammonia levels, again if severe.